class receiving students and teachers. Thank you. You may be seated. The teachers, we are still, and the students in the receiving schools, we are still on the same strand, substrand, learning outcome, and also the unit title. Now, before we go to recapping of the previous lesson, teachers, I'd like to apologize of the previous lesson. We were supposed to have, the students were supposed to have the 5 test tubes or containers filled with bases before they add the dilute acids. However, we were not able to do that, so I'd like to let you know of that. But in actual fact, acids are supposed to be added to water, not water to acids. For recapping, our first question, what is the reaction between an acid and a base cold? What is the reaction between an acid and a base cold? Receiving students, you've seen the previous lesson, so I hope you should be able to tell your teacher the answer to that question. Thank you. Let's have Livingstone.
Neutralization.
Very good, thank you. Neutralization. Our next question, you have to describe the resulting solution, meaning when neutralization took place, what solution, a new solution was produced. So what is that new solution? If I can have someone from the class, you describe the result of neutralization. Thank you. Let's have someone from Michelle's table.
Water.
Water. Or we can say the solution is neither acid nor base. Thank you. What do dilute acids and bases produce? What do dilute acids and bases produce? Let's say Roseanne, is it?
Water.
Thank you, water. I hope receiving students were able to give those answers to your teachers. Now, before we go to our topic for this lesson, I'd like us to have a look at what I have. Now, I have with me a solution. There are about 4 of them. Okay, I have 4 of them here. This is not a solution. We have it there. Okay. This solution is diluted, has been diluted. And this is a diluted ammonium hydroxide. And that's one of the laboratory bases. Ammonium hydroxide is one of the laboratory bases, and this is not the pure one. It has been diluted. So that's a sample of it. We also have another laboratory acid, okay, sulfuric acid, which has been also diluted. And that is how it looks like. Sodium hydroxide is another laboratory base. It is also, has also been diluted too. Now we expect to have something like that. The other 3 we had liquids in liquid form. We have hear more about this when we come to our lesson. This is sodium chloride. And sodium chloride is the result of hydrochloric acid plus sodium hydroxide. And the result we have sodium chloride, the salt that we normally use, some of it at home. Now, topic for this lesson, add concentrated acids and bases. In the previous lesson, we looked at adding dilute acids and bases. Now, I'd like someone to tell me what you can about concentrated acids and bases. I've shown you samples of some of them which have been diluted for our use. Someone who can tell me what you think concentrated acids and bases are. Receiving students, please, you try as much as you can to give that answer to your teacher. Where is the mic? Ella, or we can have someone from your group.
The concentrated of a solution is a measure of how much solid or solid is dissolved in the solution of water.
Very good, thank you. Thank you. Anybody else besides Malcolm? We have Livingstone's hand up, so let's have Livingstone.
Large amount of dissolved substance in a given volume.
Very good, thank you. I am not going to say whatever answer you are giving me is wrong. At least you are trying. Okay, concentrated acids and bases are acids and bases which are very strong. Very strong. They are corrosive. I believe you learned that word in the previous lesson. Corrosive meaning they can burn. While in the case of dilute acids and bases, they can be corrosive, but a lot of them, especially the ones that we get from plants, we used some of that in a previous lesson. They are acids and bases that at least have some fluid of some water in them. Okay, I'm going to go through quickly at our questions for this lesson that we are going to have. And I would like the receiving students with your teachers, if you don't have the books with you, I'd like you to get them copied into your workbooks. Now, our activity, we will be having guest speaker presentation on a video. That's not a guest speaker. And the guest speaker will be basically talking about types of concentrated acids and bases. The neutralization of concentrated acids and bases. The concentration acids and bases are actually the laboratory acids and bases. Acids and bases that are used in the laboratory that we can't use in the classrooms. And also an experiment on one common acid and base that you will view also in this video. Now, the class as well as the receiving students, I'd like you to predict quickly what you think will happen when these acids and bases are mixed together. These concentrated acids and bases, when they are mixed together, what will happen? Michelle, what do you think will happen?
There might be a reaction and color change of the liquid, because if there's too much acids, then it will take the color of the acidic substance, and if there's too much base, then it won't change and it will take the color of the base itself.
Very good, thank you. That's Michelle and her friends, that's their prediction. I hope you others also have your own predictions. So after as you view the video, you try to see what prediction you made, whether it can be confirmed in that video that you are going to view. First question, I'd like you to have that written quickly. Students in the receiving school, teachers, please, you help your students and have those questions written. What are the names of the three common laboratory A, acids, and B, bases. There are 3 acids and also 3 bases. Class, you have your books, so you should open your workbooks to the page for your activity for this lesson. The next question, you have to name the salts which are produced from these bases and acids. Now, there are different types of salts. One of those salts, we use it in our food. Besides that, there are many others. These 3 common acids and bases, they produce certain salts, so you should also observe in the video and try to see what you can get from the video. And that is A, sodium hydroxide plus hydrochloric acid. What salt do they produce? B, ammonium hydroxide and sulfuric acid. What type of salt do they produce? And C, calcium hydroxide plus nitric acid. Name the salt that this base and acid produce. Fourth question, why must acid be added slowly to water and not water to acid? There is a reason for this. The last question, you have to explain the neutralization process of concentrated acids and bases to produce salts. Now, the class and the receiving students with your teachers, you should be able to get the answers for these questions from the video that you will be viewing now.
Good morning, class. My name is Mr. Kwama. I am a lecturer here in the chemistry department at the University of Papua New Guinea. In chemistry, there are many chemicals. Chemistry is a subject that is specialized on chemicals. And we use chemicals a lot in society. In our country, Papua New Guinea, we have so many chemicals here. Much of those chemicals are made from overseas. And we import these chemicals because we have special needs of these chemicals. You go to any shop, like Food World, for example, here in Port Moresby, on the shelf, you will see chemicals on the shelf. All right. And when you go into these shelves and you see these chemicals, one subject that should come into your mind is science. Which particular one? Chemistry. Right. Chemistry, the heart of industry. Chemicals are very important to our society. One class of chemicals that we use a lot here in Papua New Guinea are acids and bases. In fact, if you want to measure how industrialized a country is, you look at the amount of acids, particularly sulfuric acids, that country uses. You go to the US and they use billions and billions of tons of sulfuric acids. That's a very industrialized country. Papua New Guinea, we use a little bit of sulfuric acid because we are not an industrialized country, really. But acids are very important. Let's begin with a definition of acids. I know this is the second lesson on these important topics of acids and bases. And I know that you may have already got a definition of acids and bases. Do I have anybody in the class, please, to tell the class the definition of an acid? Yes, please, young lady. Thank you. Sometimes we define things by what they do or what they taste like or what they feel like. That is a good definition of an acid. It tells us what an acid tastes like, bit sour, right? Anybody else, please, with another definition of an acid? Yes. It is a substance that burns you. When you touch it, it can burn you. Yes? Very good. Okay, do we have now anybody that can give us a definition of a base? A base? You can give a definition in terms of what it tastes like if you have tasted a base already. You know, sodium bicarbonate there in the shop. It sounds like it's a base.
Bases are substances that feel slippery and soapy.
Okay, that's a good definition. Base is a substance that feels slippery and soapy. In fact, one of the important ingredients of soap is a base. You need a strong base in order to make soap. So yes, okay, it can feel soapy. Thank you. Any other definitions, please, of a base? Okay, we'll take that definition. We'll take the definition of the acids as has been given to us. We'll take the definition of the base as very nice and very simple definition for young students throughout the country to remember. Okay, an acid is a substance that tastes a little bit sour if you taste an acid. Can burn you if it's a very concentrated acid. I'm using the word concentrated. I'll touch it a little bit later. And a base is a substance that feels like soap. I want to go into a more scientific definition of an acids and base. Please take note of this definition. There are basically three definitions of acids and base. In the scientific world, there are three different definitions. I will do only one. Okay? I write it on the board. There you are. An acid is a proton donor. It's a substance that donates a proton. So you need to know what a proton looks like. You need to know the meaning of a donor. You know, when you go and donate blood, you give blood. So an acid is a substance that must have a proton in order to donate it. All right? We'll give an example a little bit later. Where does the acid donate the proton to? Then the base is a proton acceptor. That's one definition of acids and base. One scientific definition of acids and base. What I'd like you to do as we finish up this demonstration is for you to actually do a neutralization reactions right there on the bench. So I'll give you the instruction and you are going to do a neutralization reaction. All right? When you have written down what you need to write down, then you can please get yourself around the burette that has been set up. Please get yourself close now so that you can see what is happening. Okay, thank you. You should now cluster around the demo or the experiment set that is been put in front of you. Firstly, in the burette, there is an acid. I think it's HCl. It's in the burette. That's that instrument on the top. That is. So the lab technicians have actually put the acid inside the burette, and inside the flask is a base. I think it's sodium hydroxide base. It's in the flask. If you get the flask and you put it underneath the tap of the burette, can you do that now, please? Thank you. Put it underneath like that. Yes, put it underneath. Get the flask that contains the sodium hydroxide underneath the tap of the burette, underneath the tap of the burette, underneath the tap, so that when you put it right underneath the tap, that's okay. Lift the tap up a little bit or tilt it, and then you can. No. Okay. There we are. Okay. Yeah, shift it up a bit. That's it. Now you are becoming real scientists. You know, scientists are people who not only talk about science, they actually do science. All right? When you are here, you are a scientist. You do science. You don't talk about it. You do it. So that is why I'm asking you to do it right now. I'll just give you a few minutes to make sure that you've got the flask right underneath the burette tap. That's right. You've got an acid in the burette, and you've got the base inside the flask. And if you turn on the tap a little bit later, don't now yet. If you turn on the tap, you're actually facilitating the acid plus a base reaction. And as soon as the tap is turned on, as soon as acid flows into the base, reactions begin, and salt is being formed, water is being formed. But you've got no way of telling when is the neutralization point. You've got no way whatsoever of telling right now. So you'll need to put in something inside to indicate to you the point at which neutralization takes place. So you need an indicator in the flask. So what you're going to do is you've got an indicator. There are two kinds of indicators that you have there, but we're going to use phenolphthalein. Phenolphthalein is the one that is white in color, and in acid it is different color. In fact, it is clear in acid, and in base it will be a different color. So as soon as your solution becomes neutralized, there will be a change in color. Color change will tell us the neutralization point of neutralization. So that's what you're going to do now. You are going to do a neutralization reaction and actually see at what point neutralization takes place. You can actually tell how much acid, because you can read it from your burette, from where you start to where you finish, you can actually see it. Okay, so what I'd like you to do is to get about 5 little drops of the phenolphthalein inside the flask that contains the sodium hydroxide, please. All right, let's do that now. So you guys use this methyl orange. Okay, you use methyl orange. Let's do different ones. Okay, one group use phenolphthalein, one use methyl orange. We've got 2 kinds of indicators here. All right. First of all, take a few drops inside the flask, the little flask there. Don't put it in the burette, no, in the flask. Yes, in the little flask. Yeah, take a few drops and put it in the little flask. That's right. This is your indicator. This will tell you exactly what point of neutralization. Yep. That's it. 1 drop, 2 drop, 3 drop, 4 drop, 5 drop. That's good enough. Thank you. All right. Okay. About 5 drops. About 5 little drops. Okay, so you guys are what? The color is yellowish, so. Okay, and yours is pink. All right. Before you do any titration, you will notice that your indicator will have a different color in the different solution. What you have there is a base, and phenolphthalein is actually pink in base, right? And methyl orange is actually a bit yellowish in base, all right? Now, when you do the titration, you will definitely see a color change. That change will indicate the point at which your acid neutralizes your base. Okay? The acid is in the burette, the base is in the flask, so you are ready now to do the neutralization reaction. Okay. You need to have a steady finger to actually do the turning the tap on. Make sure that our flask is right back underneath the tap, because we are ready now to do the neutralization. Okay, I think most people are ready. Make sure that you select someone with steady fingers. All right. So that you turn the tap very slowly so that the acid goes into the base slowly. So let's do that now. Do it very slowly. All right. Okay. Someone let the acid slowly. Another person do a little bit of shaking of the flask. Just twirl it around so that everything reacts. That's it. Okay. Yeah, and keep your eye on the burette and make sure you watch the container, watch the chains. Keep shaking, shake it, keep shaking. That's right. Keep shaking it so that all gets reacted. As soon as there is a color change, you stop. Turn the tap off. Thank you. I hope you enjoyed that little experiment. But that is basically a neutralization experiment. You have reacted an acid with the base. The acid is HCl. The base is sodium hydroxide. And you have used an indicator, basically two indicators, to help you pinpoint the actual neutralization point. The two indicators are phenolphthalein and methyl orange. Phenolphthalein, its color in acid is actually white or colorless, but in base it is pink. For methyl orange, in a base, its color is actually orange kind of color or yellowish kind of color. And in acid, it changes to more orange kind of color. Okay? So the color change tells you the point of neutralization. And in fact, if you read your burette, you will notice the amount of acid that you added from where you started to where you finished. That's the amount that neutralized the amount of base inside the flask. So what you have done is one reaction, neutralization reaction. What I'd like to finish up by saying that you always add, which one? Acid to water or water to acid? Okay, why must acid be added slowly to the water and not water to acids? Acid is always added slowly to the water. All right? You add acid to the water, not water to the acid. All right? If you have a bottle of acid sitting here on the bench, you don't get the bucket of water and start filling it in there. No, you get the acid and add it to water slowly, because you can have an explosion reaction that will take place. Basically, it's very explosive. All right. So you always add acid to water. Always in a laboratory, you must add acid to water. When you are mixing, when you are diluting your acid, right, you add the acid into the water, not the water to the acid. Right. In summary, two points I have just got across this morning. Firstly, the laboratory safety rules. Make sure you understand clearly what the laboratory safety rules are, and in laboratories always follow set procedure. And finally, the topic of acids and base. Strong acids are those acids that contains more H pluses. Weaker acids are those that contains more concentrated acids actually contains more H pluses. Less concentrated acid contains less of those H pluses. More concentrated base will contain more of the other type, the basic species, OH minus. And how concentrated an acid or a base is depends on how much of these two species that are inside the solution. Thank you very much.
Class and the receiving students, that was a video on neutralization of concentrated or laboratory acids and bases. Now we're going to go through the answers together. You have been told before the video that you are to try your best to see if you are able to answer those questions as you view the video. So we will all go through the answers together. Now, we have a look at the prediction. Michelle's prediction. Michelle, what can you say about it after you have viewed the video? What you have predicted, you and your friends?
Our prediction was that there would be a reaction when both acids and bases are being mixed together, and that there will be a reaction in change of color in both liquids.
So that what their prediction they've made, they confirm it worked. Thank you. Now in this, we've seen that the indicator we used in our previous lesson was litmus paper, because we were working with dilute acids and bases. Now in the video you've seen that they work with concentrated acids and bases. That's why they have different indicators in liquid forms that they use to see that neutralization is taking place or not. Good, thank you. Now, our second question, what are the names of the three common laboratory acids? Let's have the first one, laboratory acids. Thank you. We have Yoa.
Sulfuric acid, sodium chloride, and ammonium acid.
Okay, that's the answer who Yoa is giving. We will come to the correct answers, but I'll see what I can get from you. Okay, the second one is bases. 3 bases, common laboratory bases. Anybody who's able to give us the 3? Diane?
Sodium hydroxide, ammonium hydroxide, and calcium hydroxide.
Very good, thank you. So you should have these answers. Receiving students, you should have hydrochloric acids, nitric, and calcium. Bases, Diane has given us all of them correctly. Name the salts which are produced from these bases and acids. Now, if you were not able to get the names of the salts, when we come to the answers, you will have them. Okay, let's have a look at the next one. Sorry, this is from sodium hydroxide and hydrochloric acids. We have the salt sodium chloride. That is the salt produced. From ammonium hydroxide, that is the base, and the acid, sulfuric acid, we have the salt ammonium sulfate. From calcium hydroxide, the base, and nitric acid, we have the salt calcium nitrate. Now, why must acid be added slowly to water and not water to acid? Why must acid be added slowly to water and not water to acid? Anybody who can give us the answer? Students in the receiving school, I hope you are trying your best to give that answer. Now, this is so that there is no explosion. You have heard that in the video. If water is added to acid, there is sure to be a reaction, a strong reaction where there will be explosion. I will go through the answers quickly. Now, we have to explain the neutralization process of concentrated acids and bases to produce salts. If I can have someone to give us the answer for that question. To produce salts.
Acid plus base to H2O.
Good, H2O. And this H2O has salt in it, and it has to be heated or evaporated to get salt. Those are the answers to the first question on the different types of acids and bases. On the salts produced and the reason for adding acid to water, not water to acid, and neutralization, the process. Water and salts are produced when concentrated acids and bases are neutralized. And 2, the salt and water solution is heated or evaporated to produce salt. Neutralization of common acids and bases. We have, like Michelle has said, you add acid and base. Salt and water is produced. From that salt and water, once it's evaporated or heated, we get salt. We've already had a look at that. Our key phrase for this lesson is neutralization, and it's of concentrated acids and bases. Our next lesson is going to be on traditional dyes. Teachers, you have been reminded in a previous lesson, so I hope you've got your students to do the planning and preparation for this demonstration on traditional dyes in the next lesson. Okay, materials, you have to have all those there, as well as planning and preparation done by the students for the next lesson. And with that, thank you, class, the receiving students, for your participation.